Okay, so I am Caroline Uhler from the Department of Electrical Engineering and Computer Science at MIT. And together with Ernest Fraenkel, who is a faculty in biological engineering at MIT, we are working on causal experimentation and modeling approaches for predicting therapeutic strategies. So let me introduce the challenge in a bit more detail. So biomedical research really lacks a systematic approach to build causal models for predicting therapeutic strategies. So Ernest and I seek to develop machine learning approaches that can identify the causal mechanisms underlying a disease and thereby identify novel therapeutic strategies. In particular, our goal is to discover therapeutics that will slow or prevent neurodegeneration in ALS. Now, ALS, like many diseases, including most types of cancer, is clinically diverse and is likely to consist of distinct subtypes of the disease, each of which requires distinct therapeutics. However, unlike in better understood diseases, in ALS, the number of subtypes and their biomarkers are unknown. So this is what makes the causal inference problem really hard, because we not only want to learn the underlying disease mechanisms, but at the same time need to infer the different disease subtypes, which is critical for developing successful treatments. Now, while the methods developed here are applicable broadly to biomedical discovery, we have chosen to work on the specific disease of ALS, where we have extensive prior data and also a strong network of clinical and basic science collaborators. In particular, Ernest is the lead of the data analytics in the ANSWER ALS consortium and has access to large-scale observational data sets. So we are building on these data sets to predict the causal mechanisms underlying the disease, and we then experimentally validate our framework and also test new intervention targets by performing new perturbation experiments in Ernest's lab. Now, the space of perturbations that we could perform is so huge, so an important question is which perturbation should be performed. So in order to answer these kinds of questions, we use causality and causal inference. So we represent the underlying gene regulatory network by a directed graph where the edges represent causal relationships. Now, of course, causality is such a fundamental concept that it naturally has a long history. The research in this area, though, has mainly concentrated on observational data, a setting in which it is very difficult to infer causal relationships. But genome editing technologies like CRISPR are a real game changer and make it nowadays possible to obtain large-scale perturbational data sets. So this really changes the game for causality, allowing us to really infer some of the underlying causal mechanisms, in this case, in the disease context. So we're making use of this in order to infer the disease mechanisms and also identify new drug targets. So what have we done so far? So we have made important progress on the machine learning side to overcome the two main challenges. So one, since the number of possible interventional experiments is so huge, we had to develop a principled experimental design strategy to identify the most informative perturbations. And two, ALS is highly heterogeneous, and for most patients, actually the subtype of the disease is unknown. So we developed a causal inference strategy that can reliably identify the underlying mechanisms, even in the heterogeneous situation. So going forward, Ernest Fraenkel's lab will validate and apply these machine learning strategies in the context of ALS. And we expect that the project will lead to significant advances for ALS. And at the same time, it really also lays, and we have in this work, laid the framework that can be applied broadly to many diseases to identify targets for drug development using causal methods. So thank you very much for your attention.